Hi, good morning everyone. In this video, we are going to discuss some important MCQs for your NEET 2021 exam from microbes in human welfare chapter. Now let us start. So the first question is Monascus purpureus. Monascus purpureus is a yeast used commercially in the production of. So here they have given four statements or the four options. In out of these four, so Monascus prosperus, which is a yeast. So what the production of this Monascus purpureus in the following these four components. So the first one they have given it's a ethanol. So Monascus purpureus produces ethanol. No, it's not correct. So ethanol is produced by Saccharomyces cerevisiae. So Saccharomyces cerevisiae is used for production of ethanol. The next one they have given streptokinase. So streptokinase for removing clots from the blood vessels. So the streptokinase is produced by streptococcus. Yes, that is used for removing the blood clots. But Monascus purpureus doesn't produce this streptokinase enzyme. Next, the third statement they have given Monascus purpureus produces citric acid. No, Monascus purpureus doesn't produce this citric acid. So next one they have given that Monascus purpureus reduces the blood cholesterol. It reduces the blood cholesterols. So this Monascus purpureus this produces statins. So they produces statins, and these statins they reduces the levels of blood cholesterol. They reduces the levels of blood cholesterol by stopping the synthesis of the cholesterol. So these statins they block the pathway the or the metabolism that involves the cholesterol synthesis, so that the cholesterol levels reduce in our blood. Now let us move to the second question. The second question is which one of the following microbes forms a symbiotic associationship with plants and help them in their nutrition? Here they have given four microbes. So in these four, which microbe forms a symbiotic associationship with the plant and helps in its nutrition? So let us see the four microbes that are given here. The first one they have given Azotobacter, and the second one they have given Aspergillus, third one Glomus, fourth one Trichoderma. So these Azotobacter and Aspergillus niger that produces citric acid. That Aspergillus niger that produces citric acid, and Azotobacter it is helpful for fixing the nitrogen. And coming to this trichoderma, it forms a symbiotic associationship with the roots of the plants. You can see here the roots of the plants, and the plants and this trichoderma, both these have a symbiotic associationship. So in this, the plant get some benefit out of this associationship, and also that microbe trichoderma also gets some benefit from this symbiotic associationship. Coming to these roots, and these roots gets more amount of the minerals and water because of this trichoderma. Sorry, because of this trichoderma. So because of this trichoderma, this plant gets more nutrition and all. And next, at the same time, with the help of this plant, this trichoderma gets carbohydrates. So the right statement or the right answer for this one is trichoderma. Now let us move to the next question. So yeast is used in the production of what? So the question is here, yeast is used in the production of what? Or the question may also come like this, Saccharomyces is used in the production of what? So the statements or the options that are given for this question are citric acid and lactic acid. So the question, the options they have given here, citric acid and lactic acid, lipase and pectinase, bread and beer, cheese and butter. So we know that this yeast is also called baker's yeast because we use it for baking the for baking purpose in the bakeries. So this yeast is also used for producing the beer, that means alcohol. So you can see when we add glucose to this yeast, it makes this glucose into ethanol by metabolic pathway. So it makes that C6H12O6 into C2H5OH, and in this process we see formation of carbon dioxide, and if we add lactic acid bacteria to this rather than this yeast, what we see, the same glucose will become into lactic acid, and in this process we don't observe formation of carbon dioxide. That you have to remember. Only when ethanol is formed, only the at the time only, or along with this ethanol only in this metabolism only this carbon dioxide is formed. When lactic acid is formed, we don't observe the formation of CO2. So in this process, in big containers, we grow this yeast in this sugar extract, and this sugar becomes into ethanol by the metabolic activities of this yeast. Now let us move to the next question. And which one of the following is not an example of carrying out biological control of pests or diseases using microbes? So here they have given four options, and all these options are related to the biological control of pests. So all these four related to biological control of pests, but one is not exactly matched with it. One is not exactly matched with the its control of a disease. So now we have to find out the which one is not the example for this biological control. So the first, they have given trichoderma species, and it acts against certain plant pathogens. Yes, you can see here. So the another species of this trichoderma that is helpful for the um, reabsorption of more nutrients and minerals, like minerals, water, and from the soil. So that is a symbiotic associationship where both they get benefited. And this trichoderma species, when it is present around the roots of this plant in the soil, so the other pathogens they cannot grow in this environment. So this is a acts like a biological control in this condition. So trichoderma species, that means it is a fungus. When it is present in the vicinity of the roots, so other pathogens that cannot grow, or so that they cannot damage the plant crop. The next one is a nucleopolyhedrovirus against insects and the other arthropods. So coming to this nucleopolyhedrovirus, so this polyhedrovirus, this virus, it kills the insects. So it is belongs to the family this Bacilliviridae. So in this family, we see this nucleopolyhedrovirus group, and these members they kill the insects and other arthropods. So this virus also acts as a biological control. The third one is a Bt cotton to increase the yield. So we use the Bt cotton to kill, or not to work against the caterpillars of butterfly, so not for increasing yield. So this is not properly matched. It is not the example for the biological control. The fourth one is ladybird beetle against aphids. You can see this one. These ladybird beetles, they kills or they kills these aphids. So these aphids they take that sap from the plant, and that damages the crop yielding. So these aphids so are are killed by this ladybird. So ladybird is here a biological control. Now let us move to the next question. So which one of the following helps in absorption of phosphorus from soil by plants? So here four options are given, and in these four options one microbial organism is helpful for absorption of the phosphorus from soil by plants, so that the plant gets more phosphorus. So let us see the options that are given here. So first one, they have given the rhizobium, and second one they have given anabina, third one they have given frankia, fourth one is they have given glomus. So this rhizobium, So this rhizobium is helpful for plant for absorbing more phosphorus from the soil. So the Anabina frankia, they are useful for plant for fixing the nitrogen. Now let us see the another important question from this chapter. Secondary sewage treatment is mainly a, so what the secondary sewage treatment is. So we know that the sewage treatment is divided to two phases, the primary treatment and the secondary treatment. In some countries, even the tertiary treatment is also done. So in primary treatment, only the physiological methods are applied to separate that primary sludge. But in the secondary treatment, what happens? So in secondary treatments, so that sewage or the wastewater is treated by biological organisms. So especially the aerobic organisms are used for the treatment of that sewage water initially, so that that organic matter of that wastewater gets reduced by the actions of this aerobic bacteria. Once that is done, then this active sludge, then that activated sludge or the active sludge is further processed in anaerobic environment with the help of the anaerobic microbes. So in that process, we see the formation of CO2, we see the formation of hydrogen sulfide and also the methane. So the right statement for this question is a biological process. So we use the biological process in secondary sewage treatment. In a in a primary treatment, we see the physical process. The next question is the most common substrate used in distilleries for the production of ethanol. You know that ethanol is a very important for nowadays in industrial. We see so many manufacturers are there for producing this ethanol, and this ethanol has so many applications. And how this ethanol is produced in the distilleries or in the companies, how they manufacture this ethanol. So that means what substrate they use. So we know that when we provide the glucose, that glucose with the help of this Saccharomyces cerevisiae or yeast becomes to ethanol. And what in the which form this glucose is provided? In which form this glucose is provided for these microbes? So in the form of soya meal, or in the form of molasses, in the form of corn meal, in the form of ground gram. So in the form of molasses, we provide this glucose to microbes, so that those microbes they ferment this molasses, and they make it into ethanol. So India has become a great producer for this ethanol, and India has been producing lot of amount of this ethanol by using this molasses as a substrate. Now let us see the another question. Select statement from the following, and these statements are completely they are completely related to this sewage treatment only. So sewage treatment is very important. So in sewage treatment, we get question on the primary treatment, we get question on the secondary treatment, and also what gases are used, what gases are produced in this process. So we get question like this. So now let us see. So biogas is produced by aerobic bacteria and animal waste. So the statement is here that we know that some gases are released by this wastewater in a treatment process, and those gases are produced by the activity of aerobic bacteria. So that is the first statement they have given. The second one is the methanobacterium is an aerobic bacterium found in the rumen of cattle. So they said that methanobacterium is an aerobic bacterium which is found in the rumen of cattle. Third one is biogas, commonly called gobar gas, is a pure methane, they said. And fourth one, activated sludge sediment in settlement tanks of sewage treatment plant is a right source of aerobic bacteria. And for this question, the right statement is a fourth one. The right statement is fourth one. If you see that biogas, commonly called global gas, which doesn't contains completely the methane, we see in that biogas we see the carbon dioxide, and we also see some hydrogen sulfide. So it's not completely pure methane. And coming to the methanobacterium, yes, methanobacterium is found in the rumen of the cattle, is found in the rumen of the cattle, and where it is responsible for formation of the this biogas from that rumen of the cattle. But it's not a aerobic bacteria, it is an anaerobic bacteria. Next, let us see the first statement given for this question. So, biogas is produced by the activity of aerobic bacteria on animal waste. No. So, this biogas is produced from the animal waste only, but not using that aerobic bacteria. Aerobic bacteria cannot produce Aerobic bacteria cannot produce this bacteria like methanogens. They produce this biogas. So the right statement for this one is fourth one. So in fourth one they have given that activated sludge. So activated sludge sediments in settlement tanks of sewage treatment plant and their right source of aerobic bacteria. Yes, activated sludge is a rich source of or high source of the aerobic bacteria. That's why we use again these activated sludge as an inoculum in the aeration tanks. So if you see here, this is the primary clarifier. In the primary clarifier, we see formation of primary sludge, and then the effluent is taken into aeration tanks. In aeration tanks, air is provided to this sewage water. So in this aerated tank, we see the growth of these microbes. That means aerobic microbes, and these aerobic microbes they use this organic matter so that the organic matter in this aeration tank gets reduced in this treatment process. Then these microbes they get settled in this aeration tanks, and this then this water is transferred to this secondary clarifier. So this again is settlement tank. In this settlement tank, what happens? Settlement tank, this aerated tank contains the microbes, and those microbes are settled down. They settle down in this secondary clarifiers, and these secondary clarifiers again, that means the settle down part of this tanks they contains high microbes, and so these sludge, that means this sludge, that means the sludge that present in the secondary tank is not called the secondary sludge, it is called activated sludge. This activated sludge further that is used as an inoculum for aeration tanks. So here we have a primary tank, and from primary tank the water is that means the effluent is transferred into this aeration tank. In aeration tank, with because the air is available, the microbes, that means aerobic microbes, they use this organic matter and they grow up. After that, this the water from this aeration tank is transferred into this secondary clarifiers or the settlement tanks. In secondary clarifiers or settlement tanks, the microbes they get settled down. So like in primary tanks, in primary clarifiers, we have seen the formation of sludge with the solid substances like the soils, that is called the primary sludge. And here we see the formation of the active sludge, and this active sludge contains the more amount of the microbes. These microbes are further used as the inoculum for cla- for uh, for clarifying or for removing the organic matter from this aeration tank. So this is about this question. So now let us see which one of the, not this question is not present in our syllabus. Next, let's see which one of the following pairs is wrongly matched. Here they have given four pairs, and out of these four, three pairs are correct, and we have to now identify the wrong pair. So the first one is methanogens they have given. So methanogens produce gobar gas. Yes, they produce gobar gas, and methanogens are anaerobic bacteria that you have should. Then coming to yeast, and yeast is a fungus and which produces the ethanol. So in distilleries we use this yeast for production of ethanol, and we use molasses as a substrate. Then streptomyces, yes, streptomyces produces a streptomycin antibiotic, it is also correct. And coliforms, they produces vinegar. No, that is wrong. Coliform, it is used for measuring the how much amount of the fecal matter is present in the water. So usually these fecal matters, they are rich of this coliform bacteria. So whenever that fecal matter mixes with the water, so we use this coliforms for measuring the how much amount of that fecal matter is contaminated in that water. So this Vinegar is produced from the acetic acid only, so properly matched. So these are some of the important questions. I hope these questions are useful for you, and I hope you have understood the subject behind these questions also. Thank you for watching these videos, and thank you so much.